everyone. It's really nice to be in a similar time zone. So I don't need to wake up like 1 o'clock in the morning to share some slides or some information with our colleagues overseas. So I think this takes this great opportunity to present this topic is the tips for successful coupling of proteins to particles using AnteoBind nano-coating technology for lateral flow immunoassays and the point of care test assay development. So I will start. So in the agenda for today, we're going to quickly go through an introduction of our AnteoTech company, and then we'll discuss briefly on the industrial trends in the lateral flow assays. Also, let's touch the topic on lateral flow principles and some protein conjugation options as overview. And then we will introduce the AnteoBind technology and some physical characterizations of some during the conjugation process, like the particles behaviors, and also some functional performance of several assays as selected ones to discuss. Then we will have some conjugation conditions, the consideration, and initial scale-up procedures. And I'll start on the first one as AnteoTech. We are a company located in Brisbane, Australia. So you can see this is Australia map. We're somewhere in Queensland here, in this corner. And there's a Brisbane River across the city, so it's quite beautiful, river city. Welcome to visit us, give it a chance. And currently, our company is the ISO 9001 and aiming and coming in August, roughly August, to become ISO 13485. And our company historically has developed proprietary nanometer size binders, primers, and coatings for nanotechnology applications. So in that, because that very wide, the kind of areas of application. So currently, actually, we only have two focus on areas. Two focus area. One is energy. So our energy division now is working on the lithium battery, preparation on silicon composites and also cross-link binders. On the diagnostic and life science side is the one we're focusing on and going to talk about today is AnteoBind assay development reagents. And also we have some contract assay and conjugation services. Also, we are the suppliers to supply the raw material to the IVD companies and the point of care companies. The next slides, I'd like to have a quick discussion about the industrial trends in the lateral flow assay development. As we know, the lateral flow assay has been around for decades. It's been there for a long time. Majority of the lateral flow is used on the gold nanoparticles. It's a qualitative tests. And in that, in majority of application, it's yes or no, the kind of answers. In recent years, there are more and more the trends in called decentralization. It means that previously a lot of samples be sent to the centralized pathology laboratories, then to a process in a very big batch. And the result may take about 1 day or 2 days, and sometimes Australia even a bit longer, up to about 3 days to a week to see the results, to know what's the result and outcomes of those diagnostic tests. And the decentralization is getting more and more popular because the need, because the tendencies, and people like to know their health status. So more on site, the kind of point of need, rapid diagnostics has been start to be getting more and more popular. For example, emergency room, test for cardiac markers, and also the some markers which it needs to know the results on site and straight away, so the doctors can make some suggestions or treatment. And also there are some trends into going to the home usage of the in vitro diagnostic rapid test as well to monitor certain biomarkers or some pregnancy planning the kind of needs. And in that, there's another a trend, which is usually for example, the conventional gold-based assay is a visual like interpretation, and more like the readers based or mobile phone based, they kind of interpretation start getting more and more popular. The reason is using the reader, it reduces the human errors, and using the reader, you get some quantitative readings will be able to translate it to inform the patients or the people what needs to be done next. So usually it can be connected to a certain applications or in the cloud and certain further usage of those data. And the third one is about the gold nanoparticles. As we know, gold colloid still the mainstream because that's been existing for decades. But we can see that more and more companies are looking at other alternatives because due to some proteins or antibodies may not working very well with the gold colloid. So for example, like color lactic speeds, the different colors, the red one, the green one, or the blue ones, or the black ones. Especially recently, there are some black color lactic speeds getting popular. Or the fluorescence, fluorescence particles has been existing for a long time, but because there are so many different kinds of fluorescent kind of settings in the nanoparticles, ranging from different kinds of wavelengths of excitation and emission. And the recent emerging one, we noticed that and more people are using is called Europium particles, which is getting more and more popular. And also we observe that the qualitative test just to tell yes or no, and more test is migrating into the quantitative. They want to know what's the concentration. People want to know what's the level of the certain biomarkers in order to predict what needs to be done in the medical treatment. So that in the industry, in the several industrial trends we have observed in recent years. And if we look at the lateral flow assay, it hasn't changed for decades. It's nearly the same. As we can see that in the settings that you have a the sample pad, and the sample pad covers a little bit of the conjugate pad, and coming to the conjugate pad, then it will be a membrane, and the right hand side here is the absorption pad. And usually put in the control line and test line, so it's trap and coat your antibody here for capturing to form the immunocomplex. And on the conjugate pad here, you put in your particles for detection, either be gold, color lactex, or Europium particles. Then connect with your conjugate with your antibody, the antibodies of interest. Then coming with the analyte, and then form the immunocomplex on top of the test lines or control line. So this principle has been utilized for many years. So in that, in our hands, we also try to understand what is the most important part. And as we know, we can screen different kinds of a sample pad or conjugate pad with different kinds of buffer compositions or drying down conditions. And the membrane, we do have choices of the different membrane with different pore sizes. Size from big ones to small ones to accommodate different sizes of the conjugates. Also, we have the different kinds of striping a strategy to improve that kind of a background to reduce the noise. But I think in our hands, what we observe, a lot of these can be what we call optimized, because we can have optimized the buffer system. We can optimize the kind of membrane. We can optimize our antibody striping on the membrane with a different concentration, or co-stripe is the BSA to reduce the background, etc. But the key one, we noticed that one of the key ones is actually the conjugate. If the conjugate is not produced reproducibly, or the conjugate is cannot be made antibody retaining enough functionality, very easily you lose the assay reproducibility. It means very high CV, like one batch will be looking okay and running fine, and another batch is like a disaster. So like a percentage of a CV control is important, and also the performance. If the antibody on the particles is not functional, is not active, then you won't be able to graft enough analytes to form the immunocomplex. It means that when the analyte amount is really, really small, it's not easy to detect. So the lower limit detection, we can achieve a very good lower limit of detection. So the key components is actually what we notice is the conjugates for the lateral flow. So we want to look at like what would be the ideal particle conjugation for lateral flow developers and manufacturers. What's our ideal ones? And it would be nice to have all these characteristics in our hands in the particles we are dealing with. And as working in the industry, it would be nice like particles are pre-activated. It means that they're activated, they have a surface, like we can put it aside for a year, store in the fridge in the big batch. And when we need it, we just take it out and like remove the excess buffers and then conjugate directly to the like a protein, and that'll be like a very like a handy for us. And also we reduce the asset development time and simplify the production like schedules. If we have a pre-activated particle with a big batch and it performs the same from day 0 to day 365. Also, this particle would be nice to have a very tight particle size distribution, because if the particle, after we put a protein on, it start to like distribute like from 100 nanometer to 2000 nanometers, then there will be very like a wide kind of a size distribution. It means that when you flow into the lateral flow strip membrane, the membrane pore size is fixed. Then some of the like the particles may stuck in the membrane at the front or cannot flow properly, and the kind of signal output will be like also affected as well. And not only that, it needs a very constant and consistent surface charge. This kind of surface charge is required to retain the kind of colloidal stability. Otherwise, the particles becomes unstable, and it becomes unstable. They start to form aggregates during the storage. And the next point, it will be good to have the very consistent signals output. So it means that from the manufacturer, when the particle comes in, each time if it's a color particle or fluorescent particle, the intensity is being controlled in a certain level. So it reduces the kind of complexities when during our like developing an assay or during manufacturing. And for manufacturers, what we like want to look at also batch to batch consistency in conjugation, because one day we can make a very good batch of conjugates and perform beautifully in the strips. Then 1 week after to make another batch, we may not work as what we have expected. Signal is too low, the background is too high. Then how do we pass this batch? Because it is potentially a problem, so an issue. So batch to batch consistency and easy to scale up as well. So in R&D, we may like have a very good conjugation in a smaller 100 microliter, 1 ml. They kind of how can we scale up to be enough to make like thousands of strips or tens of thousands of strips, the kind of a conjugate amount. So also, what would be nice is particle conjugation can improve our lower limit detection and can be used both for qualitative and quantitative tests. And by the end of the day, it's a similar cost of goods to like a gold colloid. And also, there are some readers available can read these particles if it's not colorimetric based. So that's the the ideal particle conjugation for the lateral flow. Then we can quickly touch what's the conjugation options for lateral flow particles. I think for we from the colloid industry, we are very familiar with the passive absorption. To have the gold and the gold ready is a certain OD. Then we need to adjust the pH if it's required, depends on the antibodies or antigens. Then we may need to adjust the pH again for the blocking. And though it's a passive absorption for gold, but there's some preparation, and sometimes the gold is so easy in terms called crash. When it crashes and aggregates, it turns purple. then the whole batch, we may not be able to revive it. So it's like a ease of use. We just add into it, but sometimes it's not easy to control the outcome of a conjugation. And chemical covalent methods, I think a lot of people familiar with this is you have a particle which is carboxylated, then you have EDC chemistry to activate the carboxylic acid, and it's ready to bind. But as you know, as soon as you activate the carboxylate particles with EDC, you need to use it straight away. You cannot wait. You can say, oh, wait for a week, and the particle don't wait. They need to find the primary amine from the proteins or macromolecules to bind onto it straight away. So it means if the chemistry is not done like straight away, you may start losing its kind of a functionality. On the other hand, as we know, the EDC chemical, the chemical itself is like a moisture sensitive. So it's we need to be very carefully handle this chemical to ensure each time we put in our chemical covalent conjugation is the same amount, so we can control the process. But the upside of this chemical covalent is pretty stable. Yes, still some may come off, but in comparison to other methods, previous methods, it's more stable in bonding. The third one is called the binding proteins. And some proteins may die on the passive absorption or performing really well, even using the chemical covalent. We may put in the streptavidin, we may put in the protein A, protein G as another layer outside of the particles. And the streptavidin will bind to biotin antibody or biotin antigens. It's like a lot of people using this strategy when they build certain infectious disease, like flu A, flu B, or some assays. And in doing so, to reduce the kind of antibodies may not be performing really well, and it also can provide more to reduce a little bit steric hindrance, to allow the antibody binding site to stick out a bit more. But some antibodies may not work in this way, and sometimes it has its own issues as well, because also need to retain the protein A, protein G activity, the streptavidin activity on the surface for a long time. The fourth one we'd like to introduce is AnteoBind, which is an Anteo technology utilizing the metal ion polymer, which is a little bit different or very different to the previous one. It's just and the service and I'll introduce is a new alternative in your toolbox. So for example, if you try passive chemical or those binding protein methods, it's still going nowhere for your assay developments, you may consider this kind of metal ion polymers. It may help to conjugate your proteins or antibodies. So why it would be able to help? When we develop it, because it's metal ion polymer, is made by metal ion, is a positively charged complex. It's very small, it's a nanometer in size. It is an aqueous-based solution. So it's very simple to mix. It is not a harsh chemical in comparison to other methods. Also, it's quite stable, activated surfaces. When the surfaces is activated, it's ready to use, and also it maintains its stability in solution for more than a year. So it means that we have a color latex particles or Europium particles activated by the AnteoBind or metal ion polymers, and it will be stable for a year in the fridge. So anytime you can go back to find the batch and use it, it will be the same. And the binding is also another a good point I'd like to quickly show here. Like today, we have a carboxylated particles, the surface. So on the surface is a negatively charged. So it's carry negative charge on the surfaces. So usually it's a minus 30 or 40 millivolt in the zeta potential measurements using the laser scattering methods. So these particles are very, very negative charged. Then metal ion polymer comes in called AnteoBind, which is a polymer can behave just along on the surface of the carboxylic acid particles and make the negative charge suddenly become very positive charge. So this positive charge metal ion polymers will stick out their hands and ready to bind to the antibodies or proteins will comes in through multi-point coordination binding. So this multi-point coordination is either through a additive bonding or chelation or certain coordination interaction with the amino acid side chain carrying the electron donating group from the antibodies or proteins. So the antibodies or protein who have the amino acid side chain will be able to interact with this metal ion and forming the kind of additive bonding or coordination will be captured by the AnteoBind surfaces. And that the surfaces will become antibody surfaces after the blocking, etc. So this is what we have developed called AnteoBind technology. It will be just like something in the middle between the particles of choice of your interest and the antibody or the protein of a choice. The process we have found in our hands is pretty reproducible and very easy to scale up, like in our hands, in our experiences. So in that, we have actually many point of care customers who have utilized this one successfully in their asset developments. The local one in Australia here, we're a company called Illum Healthcare. Illum, a company that developing the fluorescent lateral flow assays using fluorescent quantum dot particles for infectious disease, for example, the flu A and flu B. And I believe they already near FDA approval this year. So I think that's a very good outcome. So on that front. Then we have several companies in Taiwan, in China, in Asia Pacific area. The majority of them actually is using Europium based particles for lateral flow assays for infectious disease, and fluorescent particle based for cardio markers. That's in several companies in this area. In Europe and US, we also have companies currently still evaluating for the HIV antigens and activate the active TB diagnostics. So this type of here is active TB diagnostics, and also some fluorescent particles based on microfluidic point of care for cardio markers. These customers in Europe, we already signed a supply agreement with them. So I think there are several customers in this area point of care. They use our technology to activate the particle of interest, and then already developed lateral flow test for nearly a commercialization. So what we have is for the lateral flow asset development, we have 3 kits. I think Koma will biotech will be able to elaborate a bit even more detail here, just to quickly introduce them. First one called universal coupling kit. In this kit, we have activation reagents, buffers, etc., and also up to blocking and storage, suitable for the size between 200 and 3,000 nanometers. But because this one contains some surfactants, so it's ideal for hydrophobic carboxylic acid beads. And the other one in the middle called nano kit. When we develop this one, it has activation reagents and the buffer sets for conjugation, blocking, and storage. But we targeted the size smaller for lateral flow purpose, less than 500 nanometers, carboxylated. And some customers, the customers in Europe, actually they're using the sulfate-based, not carboxylic acid-based, because it's also negative charge particles. And is suitable for color, latex, Europium, magnetic, or fluorescent particles, as long as they carry carboxylic acid as a functional group. And coming soon here, we have a pre-activated Europium particle kit. It means we selected 1 Europium particles, we pre-activated it as a kit format to provide even easier kind of a handling for the customers. So what is the advantage of AnteoBind Europium particles versus conventional? The AnteoBind, the binding technology, as we mentioned, is a patented nano-coating technology. We have a metal ion polymer based, and after activated, it's stable for a year and it's a gentle binding, so retain a lot of activities of the antibodies on the surfaces. So it reduced assay development cost because the kind of a screening of a buffers is not required anymore. And also the antibody antigen binding is more predictable. And sometimes in conventional methods, we just don't know if this antibody is going to work. So we need to test and trial several rounds, and especially the pH screening for some antibody is very important for the EDEC chemistry, and it takes time. And the detection method, because Europium particles is 300 nanometer in size. In actual size in comparison to gold, gold is small and tiny. So it means if when it forms immunocomplex, you have more like a signal intensity generated by the Europium. And also Europium signal in comparison to other fluorescent particle is much, much stronger because it stays much longer in that kind of the fluorescent intensity. So it allows it to be utilized for time resolved, the kind of fluorescent detection to reduce the kind of background or environment of fluorescent interference. And much better the kind of a signal dynamic range of the 3 or 4 logs and the kind of intensity as well. And by doing so, plus the active antibody, you can improve the lower limit detection and usually better than gold in our hands. And the third one, reproducibility, is pretty easy to scale up and show the kind of a good batch-to-batch reproducibility when it versus the conventional methods. So the particle we select to demonstrate here today is the 300 nanometer. It's under the TEM, the transmit electron microscopy image of antibody conjugated, the AnteoBind activated 300 nanometer Europium particles. So it'll be utilized in this like the conjugation. So how do we prepare it? So firstly, you need to activate the particles. So the particle is being activated by activation reagent. Then it can be stored away, like in the fridge. And then when the time you need it, you can take it out and utilize it for like a conjugation process. Firstly, you need to wash out the excess, called a particle storage solution. Then you come in with the antibody is already in the conjugation solution. You mix these two together, 1 to 1, and then you incubate it at room temperature for an hour with a gentle agitation, and the antibody will bind to the surface. Then you don't need to wash off the antibody. You can come in with your BSA for blocking, and block it for an hour. And after blocking for an hour, if it's a small scale like preparation, you centrifuge down the particles and then replace it with the storage buffer, and the particle is ready for the downstream analysis. So it seems it's like usually half a day work. Like if you have activated particle, you can prepare the conjugate, like finish the conjugation about lunchtime, and you can run the assays in the afternoon if you start from the particle already pre-activated. In these slides, we'd like to cover what we have mentioned, why the size of the particle is important for the lateral flow assay. So we have observed that when we prepare all these conjugates, if we don't have a very tight, the kind of a size distribution, then we will experience very high percentage of CV when we run the test. So we run the 2 strips or 3 strips, they give you like quite different numbers. So in that, we try to promote is we need to have very tight a size distribution when we prepare all these conjugates. So for example, in cardio, a troponin I, antibody conjugation, this commercial antibody we obtain from a commercial company. Then we conjugate to AnteoBind activate Europium particles. This is the size distribution chart. So you can see it's a very tight distribution before and after activation and conjugation. So the size is from 321, roughly in the range of the 372, and polydispersed index is extremely low, around 0.03. which is a very good sign, very good for the particle distribution. Any particle distribution less than 0.1 is regarded as very tightly distributed. Then on the surface charge is a very good indicator. The bare particles is very negative, negative 40. Then you add the AnteoBind, which is a very positive charge metal ion polymers. It become very active, become very positive. Here into the range about positive 42. Then you put antibody on, it becomes negative again. So the shift of the zeta potential surfaces measured by the laser scattering helped us to know that if the surface has been activated or not, if antibody has a binding or not, and then it gave us a very good indicators of a quality in process quality control to ensure that the particles carrying proteins is a certain range of charge along the during the storage. So that's the example physical characterization, and in the whole when we're dealing with any particle conjugation, we will be able to know what is the particle's behavior looks like before we draw it down to the conjugate pad. If we don't know exactly if any aggregates already appear, or is any a very bad or not very wide kind of a size distribution, we may experience some an unknown performance issue later on in our strips. So in this, you may see maybe just one picture. Actually, for CTNI, we repeat many times, and each time we get very, very similar results to the size distribution and the surface charge as well. We apply the same principles to all the assays we try to develop in-house as a model assay, as a demonstration. We observe the same, a very tight size distribution, and also the same trend in the surface, kind of a charge. So we talked about a lot of particle preparation, and now is the assay. These are the typical the lateral flow assay design principles. Usually you have a gold antibody, so anti-species antibody. It may come from different, it's not mouse or rabbit or can be chicken. Then on the right-hand side, the foaming sandwich is called a test line. You have the antibody to capture the biomarkers, and also you have anti-biomarker mono antibodies to form the test line, the immunocomplex. If it's a fluorescence, like Europium, you come in with a UV, and then there'll be fluorescent signals about 610 nanometers. For other particles or lattice particles, maybe using reflective methods to measure. So this is the result we obtained from the previous slides, the CTNI Europium particle conjugates in the half strips lateral flow immunoassays. So we test them in both in buffers and also in serums, and it shows that the linearity is really good, and also the percentage of CV is really tight. So because the percentage of CV is really tight, the kind of variation is low. So it means for calculate analytical a lower limit detection can easily go down to 20 picograms per ml for both assay in buffers or in serum. So in comparison to the literature being published, roughly is about 10-fold kind of improvement. And the signal noise ratio is also quite encouraging as well. Between the 1,000 picograms and the background is over 50. Sometimes you can get up to 100 to 1. So that kind of a slope and that kind of a signal noise ratio give us more confidence that using AnteoBind CTNI or Europium particles can build very sensitive CTNI assays. Then to present quickly on another two biomarkers here. One is, both of them is related to sepsis biomarkers. So what we did is we tried to be more creative. Before we only put one antibody on one particles, but in this one we put three antibodies on one particles. So we have anti-CRP, anti-PCT, and anti-IL-6. So the C-reactive protein, interleukin-6, and procalcitonin PCT, antibodies on one particles. And the design will be like very similar to what we had, just three more lines. So you have a control line on the top, then you have like three biomarker lines from PCT, IL-6 to CRP. Because CRP is quite abundant in the system. So actually what we focus on is IL-6 and the PCT. Also, we use a reader, which actually is Australian, a company, is based in Melbourne, and the C and FDA ready, that kind of a reader called Axon. And this reader has a flexibility, a very wide range of cassettes. It's a camera-based, can do quantitative or qualitative, and the optional like a barcode and scanner and the printers. And also you have a very good software for the assay developments, and we utilize this one to develop this assay to prove a concept. So on the right hand side, you can see the test line, the control line. The intensity can not only just seen as a picture, but also can be seen a little bit 3D, the kind of graphical presentation to tell what's the distribution of the signals when it's read. So using this one, we have obtained some encouraging results. For example, AnteoBind sepsis biomarkers in the full strips, we dry down the conjugates and full strip settings, etc. Using the PCT as a model assay in the 50 percent of human serum as an example, and we spike different concentration of antigens. So the x-axis is from 0 to 15 nanograms per ml. And the y-axis is the signal output of the graph here. We just only select several strips as representative, and the CRP is already abundant in the this particular human serum. So you can see that there's no cross reactivity to IL-6. At the same time, it gives quite good dose response. For the PCT, it is sitting around 50 picograms per ml, and analytical lower limit detection, which is encouraging because it's much more sensitive than gold. On the IL-6 proof of concept in the full strips sepsis test, in this multiplex test, similarly we check the IL-6 antigens with a dose response from 0 to about 1 nanograms per ml. So on the right-hand side, you can see that the background is clean, and then the more IL-6 you have, the more signal it generates. And the signal output is roughly sitting between 10 to 20 picograms per ml. It can be differentiated as an analytical lower limit detection in our observation here. So this result also is very encouraging because it's significantly better than using the conventional methods. So far, talking about this ideal particle conjugation, we did have a system can provide a particle is pre-activated. For example, all the Europium particles in the previous tests, we use it in the time of 6 months of proof of concept development. And in that is the same batch of the Europium particles. We haven't noticed any deterioration of change of intensity of the Europium particles. And it's ready to use and reduce the assay development time. But anytime you go back, you're using the same batch. And very tight particle size distribution as well, as you have demonstrated. So all the CRP or the PCT and IL-6 tests all go through the same size distribution, the kind of tests and checking, and also the surface within the control level. And you have a very good signal output and reproducible. For example, the proof of concept test, we have repeat each one 3 times, and we got the same results. 3 batch of conjugation, and the result is the same. So the result we shown previously is just 1 batch of the result. And it can improve the lower limit detection in this case, and also can be utilized to further develop into quantitative tests if it's needed. And the similar overall cost of goods, because the Europium particles is really bright. You don't need to use a lot. And usually in the case, like you use a much smaller amount. So the antibody amount is used less, and the particle is also used less. And the cost of goods will be quite similar to like a colloidal gold. And the readers are commercially available, so easy access to those readers as well. So there are some like assay component guide. I'd like to focus on between 100 and 400 nanometer in size of the particles. For example, like color, latex, or the Europium or fluorescent particles in the conjugation. If the particle is activated by the AnteoBind using the Nano-kit, for example, what is being like noticed that is the antibody amount is about 100 and 200 micrograms per milligrams of particles, but also depends on the size and depends on the antibodies as well, but it's usually the range. For separation, recommended using centrifugation for a small batch, and it also depends on the time. Sometimes you spin a little bit harder, you will needs less time. It depends on the size. If the particle is really small, you need to spin a little bit longer. The protein conjugation step, we need to avoid using the phosphate buffer EDTA or other metal chelators due to the AnteoBind services is a metal ion services. So these are metal ion chelators. Metal chelators may interfere during conjugation, but it's okay in the assay buffers when you're running the assay. The blocking and the blockers suggest use bovine like serum albumin, BSA, or casein, or other blockers you're familiar with. As long as it has electron donating group, will be able to block. Always hydrophobic blockers will block the patchy areas without the carboxylic acids. In storage in solution, phosphate buffer is not recommended. High amount of proteins is not recommended. And also some metal chelators and others in the buffer you need to avoid. And the drying buffer of a conjugate pad, we usually just put in some sugar, some lower amount of surfactants, and lower amount of BSA casing for drying. And the conjugate pad usually needs some pretreatment. And this pretreatment prefer to use a little bit higher amount of BSA and some surfactants and sugar. The running buffer needs to be tree-spaced. Phosphate is also okay, but I think tree-spaced works better. Started with 1% twin, 20, then with some other surfactants if it's required, and some sugar or a peg to even need to increase the kind of viscosity. The particle dispersity, if we experience some aggregations during the conjugation steps, we can do the bath sonication to reduce it. Also on the membrane, usually the size, the membrane, the choice between 90 and 120 HF equivalent. And the antibody usually strip at 1 milligrams per mil, can be lower or higher, depends on the need. And also can co-stripe together with the BSA to reduce the background if it's required. And the conjugate pad, usually you have plenty of a choice you can choose from. So that's the assay component guide. And before we conclude, we'd like to have initial scale-up studies we have done. What we did is activated the particles, you add directly into the protein solution and just mix. And after mixing, that'll be conjugate for an hour in room temperature. So this mechanical mixing either can be utilized like a magnetic stirrer bar or overhead stirrer if it gets bigger. So you add the antibody for an hour, then add blockers for another hour. So it's a higher concentration of blockers to put into it. Then in room temperature with a gentle agitation mixing. Then after that, you can separate by centrifugal forces at room temperature for 4 degrees. After buffer exchange, the conjugate is ready to stripe on the conjugate pad. So usually in this preparation can enough to do about 20,000 tests in a small batch that is. And because the mechanical methods can be easily scaled up.